So for our next couple talks, I'm very excited again to introduce you to some wonderful younger researchers who are doing innovative work and were selected from our abstract submission competition. The first talk that we'll hear is from Dr. Kirsten Meyer, who's a postdoctoral fellow at the University of Toronto for the time being. She is in the transitional phase of her career where she'll be taking on an independent lab very soon. And she specializes in novel antimicrobial discovery and bacterial extracellular vesicles. Today she'll tell us more about her work learning from natural bacterial extracellular vesicles to counter antimicrobial resistance mechanisms. Thanks, Kirsten.
So good morning, and I'm honored to be with you all today and to share on my research. So this is very early discovery phase research about how we can learn from nature more strategies to counter antimicrobial resistance. And I want to start with what I see as a silver lining to AMR. And this is that AMR is widespread and ancient in nature. So how do we know this? Well, on the slide, I have a phylogenetic tree of beta-lactamase enzymes. Not visible here, but perhaps visible over there. And from genetic sequencing, we now know that these beta-lactamase enzymes, which are the primary mechanism for conferring resistance to beta-lactams, are widely found in gram-positive and gram-negative bacteria of all sorts of species all around the world. And so from this, there can be a phylogenetic estimate of their age, and this puts them at 2.4 billion years old. So why is this a silver lining? Well, because it means that microbial medicine makers have had the chance to counter-evolve anti-resistance strategies. So most of the antimicrobials that we use have actually come from the specialized metabolites of Streptomyces bacteria. And these are sometimes called nature's medicine makers. And so many different types of bacteria can make different beta-lactams. So for example, Streptomyces clavularis contains all of the biosynthetic genes required to make a cephalosporin called cephamycin C. So cephamycin C, like other beta-lactams, is a very potent inhibitor of the enzymes that make peptidoglycan for the bacterial cell wall. But Streptomyces clavularis doesn't only make cephamycin C, it makes a second molecule, clavulanic acid. And clavulanic acid turns out to be a beta-lactamase inhibitor. So clavulanic acid protects cephamycin C from resistance. And many will know that clavulanic acid was developed for use in the clinic and has actually preserved beta-lactam efficacy for several decades. But on the molecular biology side, what's really fascinating is that the biosynthetic genes that are required for the production of these two molecules are actually co-localized and co-regulated together in the genome. And this is called a supercluster, and it's genetic evidence that this has evolutionarily been selected for to be produced as a combination. Despite the utility of clavulanic acid, this type of strategy has been relatively underexplored in research, and it's something that I really want to dig more into. So I was trained in pharmacology, so as I think about these natural combinations getting produced, one of the questions that I have is for this bacteria making this combination, how could they ensure joint delivery of it to another microbe? Because the traditional view is that these specialized metabolites are synthesized inside the cell and then pumped out into the environment. But I've been really intrigued by reports in the literature from recent years that sometimes metabolites can be secreted by bacteria in extracellular vesicles as an alternative secretion system. And so with my pharmacology background, my question is, are these natural drug delivery systems for the microbes? And what can we learn from them that might help us counter antimicrobial resistance? So in my research for my postdoctoral project, I began by screening a small panel of Streptomyces soil isolates for their ability to produce antimicrobial extracellular vesicles. So I cultured the Streptomyces, took the cell-free supernatant and separated it into a high molecular weight fraction that would contain extracellular vesicles and a low molecular weight fraction that would contain freely soluble antimicrobial specialized metabolites. And I found a strain, WAC276, that I'll tell you more about today, that was secreting antifungal extracellular vesicles. So the vesicle fraction from this strain is able to inhibit the growth of the pathogen Candida albicans on agar. And using electron microscopy, I could see that this fraction contained abundant lipid-bound extracellular vesicles. And to note, this was not a rare event that I found. So actually, in 9 out of 17 fractions that I looked at, Streptomyces were able to make antimicrobial extracellular vesicles. And so I purified these WAC276 extracellular vesicles, used mass spectrometry to identify that inside they packaged 2 antifungals. So one of those was the ergosterol targeting polyene candicidin, and then the second one was an electron transport chain inhibitor known as antimicin A. And very exciting to me, when I sequenced the genome of this strain, the biosynthetic genes that are required for making these molecules are co-localized together as a supercluster. So evidence that this combination may have been evolutionarily selected for. So then to look into the ability of these EVs to act as a natural drug delivery system, I've assessed their ability to deliver fluorescent small molecules to target microbes. So for example, if I label WAC276 EVs with a red fluorescent lipid dye, FM464, and then incubate the EVs with Candida albicans, the EVs are able to deliver that FM464 into the Candida albicans. I've also assessed this with EVs from a different strain, different Streptomyces, WAC288, and these EVs are able to transfer either a lipid-localized antibiotic vancomycin in the green or a hydrophilic anthracycline in the orange into the cytosol of Bacillus subtilis. So importantly, this EV delivery of molecular cargo occurs directly into the cells, countering the intrinsic resistance of the cell envelope, which is a really important mechanism of intrinsic resistance that microbes have. Okay. So as a natural drug delivery system, I'm also very interested in how these EVs might act in biofilms. So biofilms are these aggregates of microbes that are very relevant to both nature and to the clinic. So on the left is a micrograph of a Candida albicans biofilm on a urinary tract catheter. And of course, problematically, biofilms are highly tolerant to antimicrobials. But biofilms are also how most microbes are found in nature. And on the right is a biofilm of a cave slime Streptomyces isolate. So Streptomyces are growing in biofilms. Perhaps natural Streptomyces EVs might work well in the context of biofilms. And this is actually what I find. So 4 out of the 6 Streptomyces EVs that I've looked at have potent biofilm activity. And I'll go into the data for that for WAC276, candicidins, antimicins, and these extracellular vesicles. So I grew biofilms of Candida albicans and assessed the efficacy of different treatments on the biofilm metabolic viability, which is on the y-axis of the graph. And what I'm showing you first in the black is that the biofilms are tolerant to candicidin alone. So that's concentrations on the x-axis. And now I have a second axis for antimicin concentration, and on their own, antimicins are also not particularly efficacious against biofilms. But excitingly, when I put the 2 molecules together, the combination is synergistic against these biofilms. And when I compare that to these extracellular vesicles that are containing the same amounts of candicidins and antimicins, the EVs are even more potent. So these vesicles, so this evolutionarily selected for combination, as well as the vesicles, have enhanced antibiofilm mechanisms at play. And then moreover, I tested the EVs against a clinical isolate of Candida albicans that is polyene resistant. And so this data is in the open purple squares. And the vesicles maintain really good potency against this resistant isolate because of that second packaged antifungal, the antimycin A. So these EVs have both anti-intrinsic and anti-acquired resistance mechanisms. So I'll just summarize what I've been telling you about. So bacteria have evolved combination antimicrobials that can help treat resistance. They can be packaged and delivered via extracellular vesicles. And this delivery can help get antimicrobials directly into cells. They can have potent antibiofilm activity. And so if we can understand the biochemistry that's behind these effects that we see, this might give us some more strategies that we can use for human therapeutics against resistance, or animal therapeutics, or environmental needs. So I'm very fortunate to be starting a research group at the University of Waterloo to continue to do this, to investigate the biochemical mechanisms of these extracellular vesicles for antimicrobial strategies. And we'll take 3 different research directions. So one is trying to understand the mechanisms of how these EVs are actually formed, as that's not well understood. And then really to dive into what is contributing to their efficacy against resistance, and can we turn this into translational delivery approaches. So I'm looking forward to many collaborative opportunities, perhaps with you all on further understanding AMR and how we can counter it. And on the collaborative note, I want to acknowledge the support that's made this work possible, and in particular from EPIC. I have a Career Transition Award from them, and also for wonderful collaborators within the EPIC network at U of T. So thank you.